So, let's go.
We have not found life on Mars. That is the official position of every space agency on Earth, every peer-reviewed summary, every press conference held in the last 50 years. But that is saying the evidence doesn't exist. It is saying the evidence has not yet been classified as life. Those are two different things. And the gap between them, that careful, deliberate gap, is where the next 9 entries live. Number 9. Recurring slope lineae. In 2011, Lujendra Ojha, then an undergraduate at the University of Arizona, noticed something in images from NASA's Mars Reconnaissance Orbiter that nobody had formally documented: dark streaks appearing on Martian slopes in warm seasons, fading in cold ones, and returning reliably year after year. Ojha published his findings in Science in 2011, and the lines named Recurring Slope Lineae, or RSL, became one of the most contested features in Mars science. They appeared on sun-facing slopes. They flowed downhill. They were darker than the surrounding regolith, suggesting moisture. By 2015, spectral analysis confirmed hydrated salts, perchlorates, and chlorates in the streaks. NASA held a press conference. The headline was, "Liquid Water on Mars." The implication was something else entirely. What makes the RSL particularly unsettling is not the water itself, it is the pattern. These are not random. They return to the same locations, on the same slopes, within the same temperature windows every single Martian year. On Earth, there is a category of organism that exploits exactly this kind of predictable, seasonal brine environment. They are called halophiles. They don't just tolerate salt, they require it. The 2019 reassessment by Colin Dundas at the USGS suggested the streaks might be dry, granular flows, sand and dust, not liquid. But the hydrated salts remain in the spectral data. They require water to form. Something is delivering that water seasonally to the same locations year after year. And the alternative explanations, sublimation, dust devils, wind, do not account for the seasonal regularity with the same precision that a biological explanation would. The truly disturbing implication is that if something is using those brines, metabolizing them, sheltering them, timing its activity to the Martian seasons, we would have no way to distinguish its chemical signature from the inorganic chemistry we already know is there. We are not looking for a creature, we are looking for a pattern. The pattern is already there. The lines return every year. Something causes that. We have decided, for now, that something is geology. Number 8. The oxygen anomaly. In 2019, Melissa Trainor at NASA Goddard Space Flight Center published a paper in the Journal of Geophysical Research reporting that the Curiosity rover's sample analysis at Mars instrument had detected something that could not be explained: seasonal fluctuations in Martian atmospheric oxygen that no known chemical process could account for. Mars has a thin atmosphere, about 0.13% oxygen, compared to Earth's 21%. That trace amount was expected to behave predictably, following the well-understood seasonal cycling of carbon dioxide. CO2 freezes at the poles in winter, releasing other atmospheric gases proportionally. The math was known. The ratios were known, but the oxygen didn't follow the math. It rose by as much as 30% in spring and summer, then dropped in autumn. The deviation was too large, too consistent, and too regular to be a measurement error. What makes this particularly disturbing is the list of known processes that could produce it. Abiotic sources, UV breakdown of CO2, hydrogen peroxide photolysis, cannot account for a 30% swing. The only terrestrial mechanism that produces oxygen fluctuations of this scale in this pattern is biological. Photosynthesis in spring, respiration in autumn, the precise swing that living systems produce. Trainor and her team were careful. The paper does not claim biology. It states explicitly that no known chemical process can account for the data and that the source remains unknown. That is a precise scientific statement. It means the explanation has not been found. It does not mean the explanation is simple. The truly unsettling implication is that oxygen, the element we associate most closely with life on Earth, is behaving on Mars in the exact seasonal pattern that life would produce, and the scientific community's response has been to note this, publish it, and continue waiting for a better explanation. No better explanation has arrived. Trainer's 2019 paper remains unexplained. The oxygen rises every spring. It has done this for as long as we have been measuring. We are still waiting for the geology to explain it. Number 7. The blueberries. In 2004, the Opportunity rover, operated by a team at NASA's Jet Propulsion Laboratory, rolled across a flat plain in Meridiani Planum and found the surface covered in tiny gray spheres, each roughly the size of a BB pellet that the mission team nicknamed blueberries. The spheres were hematite, iron oxide, and they were everywhere. Billions of them, scattered across the surface and embedded in the layered rock below. The JPL team concluded they were concretions, mineral formations produced when water flows through rock and deposits minerals at specific points. This happens on Earth. The famous Smoky Marbles of Utah are a terrestrial example, hematite spheres formed in sandstone by ancient groundwater. The comparison seemed to close the question. What makes the blueberry distribution particularly disturbing is not the spheres themselves. It is the uniformity. Terrestrial concretions form with significant variation in size, distribution, and clustering because the chemistry of groundwater is uneven. The Meridiani blueberries are strikingly uniform. Same size, same density, same distribution across enormous areas. On Earth, that level of uniformity in mineral deposition is associated with biological mediation, specifically with microbial mats that precipitate minerals in controlled, organized patterns. In 2014, Nora Nofke at Old Dominion University published a paper in the journal Astrobiology arguing that the Opportunity images showed sedimentary structures consistent with microbially induced sedimentary structures, SIS, of the type found in Earth's oldest fossil record. The paper was peer-reviewed. It was not dismissed. It was noted, cited, and left open. The truly disturbing implication is that billions of these spheres exist on the surface, in the rock, distributed across a region the size of Oklahoma, and their distribution matches the fingerprint of life more closely than it matches the fingerprint of unmediated geochemistry. We called them blueberries. We took photographs. We moved on. They are still there. Number 6. Organics in the ancient mudstone. In 2018, Jen Eigenbrode at NASA Goddard Space Flight Center published findings in Science reporting that the Curiosity rover had detected complex organic molecules in 3.5 billion-year-old mudstone at the base of Mount. Sharp molecules that had survived on the Martian surface for longer than complex life has existed on Earth. The molecules found included thiophenes, benzene, toluene, and small carbon chains, the kinds of organic compounds that form the structural backbone of biological systems on Earth. They were found in the sheepbed mudstone member of the Murray Formation, a lakebed deposit from a period when Mars had liquid surface water. The rock had preserved them for 3.5 billion years. That preservation alone is remarkable. It means Mars's geology had been protecting organic chemistry at least since the era when Earth was producing its first microbes. What makes this discovery particularly disturbing is what it does not rule out. Organics can form abiotically, from meteorite delivery, from UV chemistry, from volcanic outgassing. The paper is careful to say so. But the specific combination of molecules found, and their preservation in lake sediment from a demonstrably habitable environment, is the same type of chemical fingerprint that precedes every finding of ancient life in Earth's geological record. We call it a biosignature precursor. It means the chemistry is correct. The question is not whether organics are present. They are. The question is whether they are remnant fragments of biology or abiotic products that mimic it. On Earth, when we find this chemistry in this context, preserved lake sediment, ancient habitable environment, complex molecular structure, we do not call it a biosignature precursor. We call it a fossil. The truly disturbing implication is that we have confirmed the molecular building blocks of life, preserved in sediment from an ancient lake, on a planet that was habitable for hundreds of millions of years, and the scientific response is to note that this is consistent with the presence of life without confirming it. The chemistry is there, the environment was there, the preservation is there, the molecules are in the rock. They have been there for 3.5 billion years, waiting. Number 5. The ALH-8401 meteorite. In 1996, David McKay at NASA's Johnson Space Center, along with colleagues including Everett Gibson and Kathy Thomas-Rassouli, published a paper in Science announcing that a Martian meteorite, cataloged as ALH-8401, recovered from the Allen Hills ice field in Antarctica in 1984, contained structures that they believed were evidence of ancient Martian microbial life. The meteorite was 4.1 billion years old, formed from Martian rock ejected by an asteroid impact 16 million years ago and fallen to Earth 13,000 years ago. Inside it, McKay's team found 4 lines of evidence: polycyclic aromatic hydrocarbons of the type produced by biological decay, carbonate globules with chemistry suggesting formation in liquid water, magnetite crystals in chains, a configuration produced on Earth almost exclusively by magnetactic bacteria and structures. McKay's team described as possible microfossils, 100 nanometers in diameter. President Clinton announced the findings from the White House lawn. The world waited for confirmation. What makes ALH-8401 particularly disturbing is not that McKay's team was wrong. It is that nobody has proven they were. Every individual line of evidence has been contested. The magnetite chains can form abiotically under specific conditions. The PAHs could be terrestrial contamination. The microfossil-like structures are at the lower size limit for cellular life. But the 4 lines of evidence together, in the same rock, in the same spatial relationship, have never been comprehensively explained without biology. McKay died in 2013 without retracting the paper. His co-author Everett Gibson continued to defend the findings. The paper has over 1,400 citations. It is not dismissed, it is unresolved, and the magnetite chains remain the most difficult feature to explain away. A 2001 study by Imre Friedman at Florida State University found that the chain geometry in ALH-8401 was statistically indistinguishable from chains produced by living magnetotactic bacteria on Earth. The truly disturbing implication is that a multi-evidence, peer-reviewed paper claiming evidence for ancient Martian life, published by senior NASA scientists, presented by the US president, investigated for nearly 30 years, has never been formally disproven. It sits in the scientific record, unresolved. The structures are still in the rock. The rock is in a drawer in Houston. We decided to wait for something more certain. We are still waiting. Number 4. The perchlorate problem. In 2008, the Phoenix lander, operated by the University of Arizona under NASA funding, confirmed something that should have rewritten the conclusion of every Mars experiment conducted before it. The Martian soil contains perchlorates, highly oxidizing compounds, at concentrations between 0.5% and 1% by weight, distributed globally across the planet's surface. Perchlorates are not unusual. They exist in some of Earth's most arid environments, including the Atacama Desert, where they are associated with some of the most extreme microbial life on the planet. On Mars, their presence was unexpected, but the implication that followed was worse than their discovery. When heated, as the Viking landers had done to every soil sample they analyzed in 1976, perchlorates react with organic compounds and combust them. They oxidize, they destroy, they leave behind chlorinated hydrocarbons as byproducts. What makes this particularly disturbing is the direct consequence for Viking. In 1976, the 2 Viking landers heated Martian soil to analyze it for organics. They found almost none. That absence was used for decades as evidence that Mars was sterile. But if the soil contains 0.5 to 1% perchlorate, then every sample Viking heated was ignited. The organics, if any existed, would have been incinerated in the analysis chamber. Vikings may not have found a dead planet. It may have destroyed the evidence of a living one in the process of looking. In 2010, Rafael Navarro Gonzalez at the National Autonomous University of Mexico demonstrated this experimentally. He took Atacama Desert soil, known to contain organics and microbial life, added perchlorate at Martian concentrations, heated it as Viking had, and ran the same analysis. The result matched Viking exactly near zero organics, chlorinated hydrocarbon byproducts. The Atacama soil appeared sterile. It was not. The truly horrifying implication is that the experiment that told us Mars was dead may have been, by the chemistry of the planet itself, designed to produce that answer regardless of what was actually in the soil. We tested for life using a method that would have returned a negative result even if life had been present. We concluded the planet was sterile. We moved on for 30 years. The perchlorates were always there. Viking heated every sample. We called it a negative result. We called it proof. Number 3, the Viking labeled release experiment. In 1976, Gil Levin, a bioengineer and NASA principal investigator, operating through his company Biospherix Inc., designed and flew an experiment aboard both Viking landers specifically intended to detect microbial metabolism in Martian soil. The labeled release experiment worked by introducing a nutrient solution, a broth of simple organic compounds marked with radioactive carbon-14, to Martian soil samples, then monitoring for the release of radioactive CO2. If microbes were present and metabolized the nutrients, they would exhale carbon-14 tagged gas. The result was unambiguous. Both landers, 6,400 kilometers apart, produced a strong, reproducible positive signal. Radioactive gas was released. When the nutrient solution was reintroduced, the signal increased. When the soil was sterilized by heat before testing, the signal disappeared. These are the 3 hallmarks of a biological response. What makes this particularly disturbing is that Gil Levin never accepted the alternative explanation. NASA's official position attributed the positive signal to hydrogen peroxide or other reactive oxidants in the soil, chemistry that could mimic metabolism without life being present. Levin spent the following 47 years systematically dismantling that explanation. In 2019, 1 year before his death, he published a paper in Frontiers in Astronomy and Space Sciences concluding that the Viking-labeled release data supported the conclusion that living microorganisms had been detected in the soil of Mars. The hydrogen peroxide explanation has a specific problem: the behavior of the signal. Reactive oxidants produce an initial spike and then decay. The Viking signal increased with additional nutrient doses and disappeared upon sterilization, exactly as a biological culture would respond, and precisely unlike the behavior of hydrogen peroxide chemistry. Levin knew this. The scientific community acknowledged it. The official position did not change. The truly disturbing implication is that the only experiment in human history specifically designed to detect microbial life on another planet returned a positive result, in both landers, in 2 separate locations, and the scientific community's response was to reclassify it as inconclusive, cite a competing explanation that its designer spent a lifetime refuting, and proceed on the assumption that Mars was sterile. Gil Levin died in 2021. His data has never been formally disproven. The canisters breathed, both of them. We decided it was chemistry, not life. We have been deciding that ever since. Number 2, the methane spikes. In 2019, Christopher Webster at NASA's Jet Propulsion Laboratory published findings in Science reporting that the Curiosity rover had detected a methane spike in Jezero Crater of 21 parts per billion, the highest concentration ever recorded on Mars, along with a repeating pattern of seasonal methane fluctuations that had been building in the data since Curiosity's landing in 2012. Methane on Mars should not exist in appreciable quantities. In Mars's oxidizing atmosphere, methane has a chemical lifetime of roughly 300 to 600 years. Whatever is producing it must be doing so continuously and recently. The seasonal pattern, methane rising in warm months, falling in cold ones, mirrors the behavior of Earth's atmospheric methane in one precise way: it tracks temperature. On Earth, temperature-dependent methane production has one dominant source. It is called methanogenesis. It is performed by microorganisms. What makes the methane signal particularly disturbing is the specificity of the seasonal pattern. A purely geological source, serpentinization, volcanism, ancient clathrate release, would not produce clean seasonal cycling that tracks the Martian temperature curve. Geological processes do not have seasons. They do not pause in winter and resume in spring. They leak at the rate of the chemistry, not the rate of the temperature. Biology does. The European Space Agency's Mars Express orbiter and the ExoMars Trace Gas Orbiter have provided contradictory methane readings, some confirming surface spikes, some detecting nothing. The scientific community has used this inconsistency to maintain uncertainty. But inconsistency in orbital detection of a trace gas is expected if the source is localized, if it is venting from specific subsurface locations where temperatures permit liquid water and active chemistry. Inconsistent detection is consistent with a biological source. It is not consistent with a uniform geological one. The truly disturbing implication is that we have a gas on Mars that requires continuous production, that fluctuates seasonally with temperature, that appears in localized surface readings too inconsistent for a diffuse geological source, and that is produced on Earth almost entirely by living organisms, and the official position is that we cannot determine the source. That is accurate. It is also a description of evidence that, on Earth, would prompt a very different conversation. Something on Mars is exhaling methane seasonally, in rhythm with the temperature. We are still deciding what kind of something that is. Number 1, the Jezero biosignatures. In 2022 and 2023, a team including geobiologists from NASA's Jet Propulsion Laboratory, Caltech, and the Natural History Museum London, working with Perseverance rover data collected from the Jezero Crater floor, began documenting a series of rock textures bearing precise morphological resemblance to biosignatures in Earth's earliest geological record. Jezero Crater was chosen as Perseverance's landing site because orbital data indicated it was an ancient river delta, a lakebed environment where, on Earth, life would almost certainly have concentrated, fossilized, and left chemical traces. What Perseverance found exceeded the morphological prediction. The rover documented textures that mission scientists informally named leopard rock, pale nodules on dark gray stone, and popcorn features, small, regularly spaced protrusions with an architecture that closely matched ancient stromatolites and microbially induced textural structures found in Western Australia's Pilbara region, where Earth's oldest confirmed biosignatures are dated at 3.5 billion years. The rocks also showed anomalously high phosphate concentrations, the element most strongly associated with biological systems, in spatial distributions that do not match abiotic phosphate deposition. What makes the Jezero findings particularly disturbing is the context in which the comparison was made. The scientists drawing parallels to the Pilbara are not science communicators. They are geobiologists, the specific researchers who study the boundary between geology and biology in Earth's oldest rocks. When they invoke the Pilbara, they are applying the most rigorous comparative framework available. They are not speculating. They are recognizing a pattern they have spent careers learning to read. The Perseverance samples have not been returned to Earth. That is the single obstacle between these textures and a definitive analysis. The Mars sample return mission, which would bring those cores back for laboratory examination, has faced significant budget cuts since 2024, and its timeline has slipped from 2033 to unknown. The samples are sealed in titanium tubes on the Martian surface. The instruments capable of determining whether the textures are biological are on Earth. The two sets of objects are 140 million miles apart. The truly disturbing implication is not that we may have found evidence of life in Jezero Crater. It is that we may be choosing, through budget decisions, through the slow machinery of mission planning, through the institutional caution that has defined every Mars finding before this one, not to find out. The samples are there, the analysis is possible. We have simply not yet committed to doing it within any defined time frame. The rocks are sealed in tubes on a crater floor. The scientists who know how to read them are here. That distance, 140 million miles, is the only thing standing between us and an answer. We have decided, for now, not to close it. If you want to see more videos like this, click the video on screen now and make sure to subscribe.